is my pleasure. Dr. Dominic, thank you very much for, absolutely, thank you very much for the invitation. And the introduction is very nice. So it's wonderful to be back in a place like Richmond. It's a beautiful place, beautiful setting. Tremendous facility here, which being gone 20 years is a lot has changed, and it's changed for the better. And I'm very impressed with the science here, and the faculty. I had a wonderful day. So thank you very much. And this is an incredible honor to be standing here in front of Dr. Powell. I had Dr. Powell as a student, or I was a student of Dr. Powell's in 1988.
85, 86.
85, 86. Sharper than me. And yes, and so first of all, Dr. Powell, if you'd like to say something, it's an honor for me to be here in your presence, and if you have any words, that'd be wonderful.
What?
Dr. Powell, stand up. Yeah. If you want to say something.
Could I do it after you finish?
Absolutely. Yes, sir. Is that what you want? So I had Dr. Powell for analytical, and I remember that quite well. It got me introduced to chemistry. That was, I believe, my sophomore year. And I also want to thank the other faculty here that had a great influence on me. Dr. Dominic, of course, Dr. Goldman, Dr. Myers, and Dr. Clow, who I don't think made it tonight. And I can't go on without saying thanks to Dr. Topham, Dr. Richard Topham, who, as mentioned, I started my undergraduate research with. He gave me the opportunity to work in his lab, and that really jump-started me to wanting a career in science. So we all miss him very much, and wish that he were here. So again, it's an honor to be here, and that's for the spiders. I still follow the spiders, love the spiders, and I'm very proud to be back here, especially in this setting at the Powell lecture. So now I was thinking about this a little bit, and this is why I said 88. 88 was the first Dr. Powell lecture, and that was my junior year, I believe. Is that right, Dr. Powell? I think it was.
Well, it took him some time to raise money for the lecture, and I'm happy to say that he raised enough money so that this not only pays for lectures here, but additional lectures that you have through the year.
It's a wonderful, wonderful event. And that first year, I remember it quite well. And I'm also honored to have a former classmate of mine, Bill Lacey, here as well, and my wife. I met her at Richmond in the back. And we were thinking about this, and I remember that being a pretty special day. I remember the faculty drumming up this Powell lectureship in honor of Dr. Powell recently retiring. And that year it was Dr. Gary Heathfield from the Indiana University. And I remember we were encouraged to go to that lecture. And I also wanted to say thanks to the students for coming out tonight on Friday night. It's wonderful for you to be here, and I hope it's of value to you. With this particular lecture, I remember I had a conflict. We had a conflict, and I remember this quite well. That was also the same night that the Spiders were in the first round of the NCAAs. And they, in fact, coincidentally played Indiana. I don't think this came up during the lecture. And we ended up beating them. They were a 4 seed, defending national champions. We were 13 seed. And we took care of Indiana University, led by Bob Knight. And some of my favorite Richmond basketball players were in this game: Steve Kratzer, Rodney Rice, Peter Woolfolk, and Kenny Atkinson, and led by Dick Tarrant. So I remember the dilemma: should I go to the lecture, as I'm encouraged to, or do I go to the game and celebrate afterwards? And I'm pretty sure I made the right choice. I'm pretty sure I went to this first lecture. So it's I'm pretty sure. So thank you all for attending. I think I used the word intimidating today. It's intimidating for me to be here with this incredible list of Powell lectures. But again, as an alumni and to be here, I'm honored to be here, and I appreciate the invitation very much. So thank you. So what I'm going to talk to you about tonight is some of our work, and this came up in talking with Dr. Dominy, in being in industry. Sometimes things don't last, research projects don't last for a long time, but we've been fortunate here that we've been working on nuclear hormone receptors, or nuclear receptors, for close to between 8 and 10 years, some longer than even me that have been at GSK. So it's really offered us the opportunity to do some basic research and publish some papers and interact with the academic community in a way that we're very thankful for, in addition to the drive for coming with new therapies. So just to introduce the overall concept of nuclear receptor modulation, I can't do it any better than this. I read this a number of years ago. It's a book called The Touchstone of Life, and this is a review by Werner Lowenstein in CNE News. And it just eloquently describes the kind of things that we try to learn about and appreciate about nuclear receptors in terms of modulating them for ultimately combating certain diseases. So that nuclear hormone receptor is a sophisticated relay obeying a ciphered command. The protein changes conformation when the steroid binds to it. In effect, the steroid has reformatted that protein into an altered surface where it can now bind multiplex adapters. And we'll talk a little bit about what these multiplex adapters are. Ultimately, that complex controls the transcription of DNA. And essentially, this is, and I love this quote, the steroid is bringing forth this information like a quarterback calling a play for a football team. Okay? And us as biochemists and chemists, what we like to try to do and think about this class of receptors is that chemistry offers us the freedom of molecular design to modulate these receptors in a way which we can control, but given the constraints that are imposed by the cellular information that pre-exists. Okay? So, for those of you not familiar with nuclear receptors, this is just an introduction slide to get us going. There are 48 of these receptors in humans. And they all have roughly the same globular containing structure. There's an AF1, activation function 1 domain. I'm not going to talk about that today. There's a DNA binding domain, which classically is a zinc finger. And you can see here these two zinc fingers dimerize to recognize certain response elements on the promoter regions of certain genes. And this is the ligand binding domain, which of course, as chemists and biochemists, we're most interested in. And we'll get into that some in the talk. This is a dimer of a classic nuclear receptor, LBD, or ligand binding domain. So 48 receptors. What I'm going to talk a little bit about are some examples. This is the subclass of receptors known as the steroid receptor superfamily. The receptor for testosterone, the androgen receptor, the receptor for estradiol, the ERs, the estrogen receptors, the receptor for cortisol, the glucocorticoid receptor, the receptor for mineralocorticoid, MR, and the receptor for progesterone, PR. So we spend a lot of time with these. There are drugs, we'll get to this a little bit, that bind these receptors and are very useful in the medical community. But we're interested in finding new ones that could be of even better benefit with less side effects. There's another class, or a category of these receptors called orphan receptors. These are receptors that were cloned basically by stringency hybridization to the DVD that are orphans. Either they were initially found to have no ligand, or it took a while after cloning to find that ligand. And I'll tell you a little bit about, and we've worked at GSK on a number of these receptors, but I'll tell you a little bit at the end about some work on ER alpha, estrogen receptor alpha, and reverb alpha. So taking a look at the big picture here about what these nuclear receptors do in terms of regulating physiology, this is taken from the NERSO website, a conglomerate of folks that work on nuclear receptors. And you can see that with this phylogeny type chart here, each of these receptors is coded by the type of ligand that regulates it. So again, you have steroids, you have vitamins, you have fatty acids, cholesterol and bile acids, phospholipids, heme, which I'll get to at the end, and xenosensors. Some receptors have been found to have no pocket to date, although there's a good chance that they do exist, they just haven't been discovered yet. And you can see all the different functions in physiology that are controlled by these ligand hormone, or these ligand receptor interactions: reproduction, central nervous system, lipid metabolism, energy homeostasis, bile acids and clearance of toxic compounds, xenobiotic metabolism, and steroidogenesis. So you can see they're really essential for life, back to the touchstone of life, and in terms of therapy, they're also good drug targets. But the problem with these natural ligands is that often these tools, either early on tools that we discover or the natural ligands, are really poor tools in terms of their solubility, their cell penetrance, and other characteristics that make them difficult to use for cell biology and pharmacology experiments. So that's one of our goals, and I'll get to that at the end as well, is to provide good quality tools that can help target validation and further understanding the pathways that each of these receptors controls. So for those of you not familiar with how nuclear receptors work, this is something I made, a very basic schematic, taking a lot of information and showing it in one generalized mechanism here. Typically the steroid, this comes from a lot of literature as well, the steroid hormones and other nuclear receptors are contained in the cytoplasm of a cell, typically of a target cell. Typically they're in complex with heat shock proteins. Upon ligand diffusion through the membrane to bind to the receptor, it then translocates to the nucleus, where it dimerizes, either homodimerizes or heterodimerizes, and interacts with a series of coactivator or co-repressor proteins. These are those multiplex adapter proteins that we talked about. So it's this LXXLL motif that was shown by Malcolm Parker and company that drives predominantly the binding of these large coactivator proteins to the liganded nuclear hormone receptor or nuclear receptor and drives transcription. And it can either up or down regulate genes when that complex binds to the promoter region. There's also another set of pathways that nuclear receptors can interfere with and block, and one example is the NF-kappa B pathway, a very important pathway in inflammatory diseases. So another complication is that a lot of these receptors interact with kinases, phosphatases associated with membranes themselves, or membrane receptors, and of course heat shock proteins. So the overall, one of the reasons I want to show this slide is that it really shows you the importance of ligand-driven protein-protein interactions. And I'll get onto that with some of our assays in the latter part of the talk. Again, I mentioned that these receptors are well suited for drug discovery. A number of drugs on the market. The top panel here shows drugs that bind to steroid hormone receptors. You may recognize some of these: Avista, Casodex, mifepristone, aldactone, and Flonase, one of GSK's products for allergy and allergic rhinitis. The thing about these, there are, as you may know, there are opportunities for new therapeutics. Some of these are not particularly very safe molecules. They have some debilitating side effects, and our goal is to try and retain those benefits but reduce the side effects and make it safer, more efficacious drug. How are we doing this? With modern day technology, we're using structure-guided design, crystallography, and new assays that can measure receptor modulation and help us parse through large numbers of compounds quite quickly. So to work with these receptors, typically we focus on the ligand binding domain, the full-length receptors. We've had some success with some receptors, as have others in the field. But we typically just use this ligand binding domain, this LBD domain here. There's a lot of work that's gone on with all these receptors, but I'm going to focus on the next few slides on GR. As Dr. Dominy mentioned, we had a big effort on GR a number of years ago, and it was a focus throughout the world, really, as a key drug target. It's known to be, of course, back from the 50s, the receptor for cortisol and other potent GR agonists. But amazingly, it was one of the first receptors discovered. The crystal structure had not been solved, and the protein hadn't even been purified at that point. Only very crude preps from natural sources. So we used recombinant technology to make a few constructs for GR. And we're really after really getting crystal structures like we already had for AR, PR and MR in collaboration with the crystallography group. And at that time, as I mentioned, GR was a franchise program at GSK. We had a number of products and a number of chemistry efforts targeting GR, but with no real good in vitro assays or crystallography efforts. And as I said, this was going on all around the world, both in industry and in the academic community as well. And again, it's a very important, well-known therapeutic target, such as one of the most prescribed medications in the medical community, prednisolone, prednisone, and dexamethasone. So what happened is we were working on this probably about a year or so without much effort, and there were only a couple of folks really willing to try. There was, because it's been tried in the academic community. But it really took a team effort, as do a lot of things at a place like GSK, to make this work. And one of the things that we did was look at sequence alignment. And as you can see, one particular position really showed this dramatic change between having, where most receptors have a polar residue at this position, 602, GR contained a phenylalanine. We just thought that was interesting. But then that really came to light and became more interesting. We became more interested in that particular position due to an old paper that we just happened on through a series of discussions from Keith Yamamoto. We actually hadn't caught it from the literature directly. And what's buried in that paper is this was, Keith Yamamoto is one of the leaders in the field at UCSF, leaders in the field on GR. And what's buried in there, this was a paper where they were using a random mutagenesis technique in yeast and looking at gain-of-function mutations, okay? They found this one mutation. They're using the rat receptor. Rat receptor 620, the phenylalanine mutation to serine, produced a drastic increase in receptor activity. So this could either be due to affecting the ligand binding of that receptor or affecting the amount of receptor of that mutant in the cell. And we thought maybe the second situation was perhaps more likely, given that this probably wasn't going to be near the ligand binding pocket. Tried a few experiments, and rather quickly, Randy Bledsoe should be credited for this in our lab at GSK. This one particular construct, GR, feeds 602 to serine with a GST, glutathione transferase tag, with a high-affinity ligand, produced a very nice amount of protein on the simple Comati gel here, compared to, for example, this is the same construct without dexamethasone. Okay, so it's this fusion protein that eventually allowed us to cleave off the GST, get enough of that purified GR to solve the crystal structure. And this is the crystal structure of GR. It's not, as we expected, not dramatically different than many of the other receptors in the subfamily of NRs. But I'll get to some of the differences in a few minutes, or some of the interesting things we found about it. Just to lay the landscape here for how these LBD domains are laid out. This is the N-terminus, helix 1. This is helix 3. And that forms the front face of the, as we look at it, the view we look at it, the front face of the domain. The back domain is essentially a 3 helical sandwich. The back layer is helix 7 and helix 10. And then there are 3 helices that kind of cross the top part of that domain, leaving a space in the bottom for the hormone, in this case, dexamethasone. Another key feature about these receptors is the AF2 helix, this activation function 2 helix, and this will become evident in how ligand controls the features of this AF2 helix throughout the talk. This is an LXXL amphipathic helix peptide from a TIF2 coactivator that was required in this case to get good crystals, to solve the crystal structure. You can see the orientation of DEX. The 3-oxo position is pointed back in the back part of the receptor, where the D ring is pointed toward the AF helix. Okay? Now, interestingly, when you look at where that serine is, and we tried to understand why exactly that phenylalanine to serine mutation gave us an increase in protein. And it's really quite interesting. We did speculate, based on some modeling, that it would probably be directed away from the hormone, and that's indeed the case. But you can see there's an intricate network of hydrogen bonds from some 3 embedded waters, forming a hydrogen bond with other serines, a histidine, and also backbone carbonyl, and this tyrosine. We think that really is what's happening here, is that this serine allows this receptor to become more compact, less dynamic, and express, and we can purify it and crystallize it. So again, quite a fortuitous series of events. And now practically every lab that expresses this protein uses either the Phe 602S or the Phe 602 tyrosine mutant, which also works quite well. So taking a closer look at the dexamethasone binding pocket, interestingly, and as we see with other hormone receptors, almost all of the surrounding pocket is hydrophobic, and we knew that from other receptors. But every single hydrophilic atom in the hormone is accounted for by an electrostatic or hydrogen bond interaction. Importantly, this charged clamp at the 3-oxy position from this Glen 570 and ARG 611. This hydroxyl at position C11 makes a very nice tight hydrogen bond with this asparaginine 564, which also coordinates with the 17 beta position. And the 17 alpha position back here, hydrogen bonds with this Glen 642. If we turn it around and look at the D ring, you can see this 2.7 angstrom hydrogen bond. Okay, so that explained how Dex binds. But what's interesting is we had a pretty intensive chemistry program, both with steroidals and non-steroidals, on GR, and a number of compounds that have larger substituents, particularly at the 17 beta position, were found to be very potent binders to glucocorticoid receptor. This is fluticasone propionate. This is the active component of Flonase. So we were curious, given that hydrogen bond and the apparent importance of that hydrogen bond in docking the ligand in the receptor, what actually happens to this region in the receptor to accommodate this propionate group. Okay? So what I've got here is, I don't know if people here use Pymol or Rigimol, but we found it's in some cases to be a really nice way to show some of these changes in structures. And what this is done by some Warren Delano, a good friend of mine in California, helped us with this at the time. He's the developer of Pymol, and he has this add-on called Rigimol, where he can take 2 sets of crystal coordinates and then morph in real time and predict where amino acids or side chains or other positions in the coordinates would change, going from one set of coordinates to another. Okay, and he makes these Rigimol movies, and he helps us out with these, and I think they're quite informative. We might want to turn the lights down a little bit if we can. It might help to show some of the details. If not, that's okay. So I can start this. So we're going from dexamethasone to the crystal structure with fluticasone propionate. And just to get you oriented here, again, this is the AF helix. The front face, helix 1, helix 3. This is now fluticasone propionate. Going back to dexamethasone. And you can see not much changes. The overall C-alpha backbone is relatively stable. If we look at it head on here, again, here's the 17 alpha position, and this is the overall pocket of the receptor. And you can see there's a little bit more space around dexamethasone. It's not accommodated for by the ligand. You can see when we flip to fluticasone propionate, you can see where that propionate group extends beyond where the 17 alpha hydroxyl extends. But again, not much overall change. Just pointing out where some of the key residues are around this D ring of dexamethasone. Again, here's the 17 beta position. This is the 17 alpha hydroxyl. This is the tyrosine 735 that packs up close against the steroid. Here's that Glen 64, which makes a hydrogen bond with the hydroxyl. And 3 chemothianines shown here at the base of the pocket. And there's also, just for orientation's sake, the C3 carbonyl. So if you take a look at what happens to these side chains with these Rigimol movies, it's really quite dramatic. You see this tyrosine rotates out of the way a little bit. These 3 methionines, really flexible side chains, rotate out of the way. This Glen 642 rotates out to make room for that fluticasone propionate group. And so what that demonstrates, and we've seen this with other crystal structures, and this is just one example I picked out, there are even larger 17-alpha substituents. For example, this fluticasone furoate, which is the active component in Veramist, where we've really seen even more dramatic movements of residues and even more C-alpha backbone movements when those positions are even larger. And with some non-steroidals, we can even extend all the way up to what's called the meta-channel and make an electrostatic interaction even beyond that back charge clamp. So really quite remarkable that the overall point is that these are adaptable, flexible regions in NR LBDs and really couldn't be predicted to accommodate such large ligands. It really shows you how the plasticity of the LBD can really accommodate ligands of different with different substituents, different moieties. Well, that's all good. Of course, what happens here is that ligand properties ultimately influence the nuclear receptor in the downstream pharmacology. Okay. So structures can help us designing, putting small groups in certain places in the receptor to fill space, help us to improve affinity, potency of that molecule, and drive head group, what we call head group diversity. That's the area of the hormone or the molecule that sticks out toward that AF helix. But what we don't know, didn't have a good way to do, excuse me, or to do around this time is really come up with a way to probe how those ligands can alter the conformation of that receptor and assay that receptor in a kind of a medium or high throughput way. So we were interested in developing technologies that could be used to screen large compound sets, and ultimately, because I showed you how complex these receptors are, simplify the inherent biological complexity, the signal transduction network of these receptors with a simple, robust in vitro type assay. Oh yeah, one more Powell movie here. We looked at the inside of the receptor and how ligands can influence regions inside the receptor. This is a movie Ward made for us that shows what happens on the outside with a more dramatic compound change going from, in this case, this is the estrogen receptor alpha, going from estradiol to a 4-hydroxytamoxifen. We're starting with estradiol. Again, here's the AF helix. This is the coactivator peptide, and we're gonna morph into the structure of ER alpha with 4-hydroxytamoxifen. So there's 4-hydroxytamoxifen. Here's this tertiary amine group right here that points out toward this AF helix and the loop before AF helix, and it distorts that region of the receptor and forces the AF helix into a different position as shown by the crystal structure, which ultimately knocks off that coactivator peptide and displaces it. So we knew that, and we knew that a variety of ligands can influence even more dramatically the dynamics of the outer parts of the receptor. We developed this method using a very nice system using Luminex fluorescent beads. And this is a system where we purchased these from the Luminex Corporation. They offer up to 100 different beads. Okay, each bead has a mixture of a red and an infrared dye. Two different dyes. They're simply polystyrene spheres that are injected with these two different colored dyes, each in a different ratio. So there's 100 different populations. Each population has a unique fluorescent code. We can decode that with flow cytometry. We can take each individual microsphere and couple peptides to it. Our peptides either come from coactivators themselves. We had a big effort using random peptide phase display, where you can find specific peptides to specific receptors and also specific receptor ligand complexes. Okay, we take this entire battery of peptides, make up to, in some cases, over 80. We use over 80 sometimes, different peptides coded on different 80 fluorescence microspheres. Then take the LBD and label it with a different fluorophore, in this case usually Alexa. Then add the compound, and then ask which of these beads does this receptor get recruited to. This would be a classic agonist compound, where the receptor is recruited to that peptide, And in the case where there are no or less receptor bound, then in the absence of compound, that would be a repressive situation. Okay? So flow cytometry is used to decode this and detect whether the receptor is bound. And this looks a lot more complicated than it really is. It's really quite simple assay. What it is, this is now ER alpha, an example from ER alpha with estradiol. We have on the x-axis here each of our individual microspheres with different peptides. We subtracted basal. So with estradiol, a number of these peptides get recruited. The receptor gets recruited to those peptides on the beads. In the case of a classic antagonist, similar to tamoxifen, this is raloxifene, the receptor gets de-recruited to these peptides. And you can see this little bit of change over here. What we do is we take thousands of compounds in some cases and screen them through this assay and come up with a number of these different peptide profiles, okay? It's a lot of data that can be generated quite quickly. So we use principal components analysis to reduce the number of peptides down to a smaller set, those peptides that only give us the most valuable information, and a clustering routine, and come up with, depending on how you do the analysis, a number of these different clusters. So here's the case where we've clustered these profiles into 12 different clusters. Here's estradiol and also diethylstilbestrol appear in cluster 3, classic agonists. Here is 4-hydroxytamoxifen, which I also should mention, as you may know, one of the first line treatments for breast cancer as an ER alpha antagonist. Raloxifene falls in a slightly different category. And here's one of our early unique ER alpha modulators called 7604. And it's got this acrylic acid head group. So obviously a different head group than raloxifene or the tertiary mean tamoxifen head group. But what interested us is this cluster. We had different reasons for going into this cluster, but there's a number of compounds here that were kind of passive compounds. They didn't activate and they didn't inactivate the receptor. They were relatively neutral in terms of their peptide profile. We then spent a lot of time with other assays in this area, and this is one example that we used to show that this 802 molecule is different than tamoxifen and different than estradiol. This is a simple Western blot experiment looking at how the ligand can stabilize the receptor in a cell or destabilize it, send it to a degradation pathway. It's known from literature that estradiol downregulates the receptor. This is an MCF7 breast cancer cell line. 4-hydroxytamoxifen stabilizes it, and you can see that 802 is a little more like estradiol than it is compared to tamoxifen. So to cut to the chase here, and there's a lot more data to support this that I'm not going to go into today, but I'm willing to talk about any of this if there's interest. 802 has been in the clinic. It's a high affinity ER binder. It's got this novel peptide profile, which really helped us to understand that it was different than the existing ER alpha modulators. It has a number of different cell line assays that look promising. Of course, you don't know until you go to the clinic, but the phase 1 trial shows that it's safe on the uterus, doesn't exacerbate hot flash like raloxifene does. And based on some of our data too, although the trial is not set up to look at this, we think there's a possible utility for this kind of a unique ER alpha modulator for tamoxifen-resistant breast cancer. So that's it for the steroid receptors. To finish up here, I'm going to talk a little bit about some of our more recent work, some of which is unpublished, on a couple of orphans. One is that we've been interested in the past year or two is this receptor called the estrogen-related receptor alpha. It was cloned a number of years ago, and through some work by Vincent Guiger and Ron Evans and some others in the field, it's been shown to be, and I'll get to this in a little bit, a critical regulator of metabolic function. But what was sort of uninteresting to us originally is that when we saw the original crystal structure, we saw that the AF2 is in the agonist conformation, no big surprise there, but the ligand binding pocket was extremely small. In fact, it could only, and this was, actually the original structure was solved by Novartis in Switzerland. And what they showed, and our subsequent structure showed as well, you can only possibly fit about 4 carbon atoms in that pocket, a very small pocket, and it was even deemed to be probably intractable, chemically intractable. But we took our chances with that, even though there's no natural known ligand. And the reason we took our chances is because it's known from our data and some others in the literature that it's a very important and potent regulator of energy metabolism, particularly in tissues such as skeletal muscle and the kidney. And there's very high biological validation for it as being a possible way to treat certain metabolic diseases, such as obesity and diabetes, and also some indication that it's also a cancer biomarker. So that gives us the opportunity that maybe there's a chance to ameliorate some cancers, types of cancers with this with molecules against this receptor. The other interesting thing about this is that it's also completely conserved throughout species, going back even to very simple organisms, and it falls in the same family as a steroid receptor subfamily. So taking all this into consideration, it's obviously a very important receptor. We just need to figure out a way to modulate it and get at it from a chemistry perspective. So what we did, like most big pharma, we took the approach of a high throughput screen, in addition to some other exercises as well, but the high throughput screen led us to this molecule, GSK 609. The crystal structure was solved by a crystallography group and shows that, of course, as expected, the molecule binds in the lower part of the domain. And what happens is that helix 3 and the the helix 3 region here, the helix 10 region, and the AF helix rearrangement, this is now in the antagonist conformation, that breathing, that expansion, makes room for that ligand at the base of the receptor. This naphthol fragment is positioned toward this, as the steroid receptor is used as classic charge clamp, but obviously there's nothing to charge clamp to. This phenylalanine here rotates out of the way, and there's a very important hydrogen bond between the indole NH and the backbone carbonyl alanine 300. We also used, in this case, very nice to see that you have calorimetry here. I think it's a great technique when it works, and this happens to be a very good system for us using ER alpha LBD with some of these ligands. This is an example with 609. And this technique measures heat of binding upon ligand association. If you do substoichiometrics, this is with receptor in solution, in the cell, and a solution of compound in the syringe. If you inject a substoichiometric amount of that compound into the receptor, you get a heat, in this case heat released, re-equilibration back to baseline, another injection, and consequent injections until you reach saturation. This is the binding isotherm. What you get out of this is the enthalpy of the overall interaction. You get the affinity constant, and you also get the stoichiometry. In this case, we're lucky to see one-to-one binding, as you would expect for a nuclear receptor ligand interaction. But what's interesting, and the overall point I want to make here about ligand modulation of these receptors, is that the thermodynamic fingerprint is vastly different. This is now the fingerprint for 609. The directly measured enthalpic term, blue is delta G. This is the entropic term, described as minus T delta S. In this case, 609's got a very favorable enthalpy, but an unfavorable entropy. If you look at other ligands in the same class, some of which are in the same class, you see that the fingerprint switches from an enthalpic-driven event to more of a balanced, positive entropic, positive enthalpic event. So again, what this shows is that different ligands can modulate and bind with different energetic contributions, and we think that this kind of thing, just like the peptide profiling, this kind of modulation and figuring out how to modulate receptors is relevant to downstream pharmacology when this interaction happens in the context of a cell environment. So we then undertook a series, I'm going to show you just a snippet here, a series of cell-based assays to try and figure out what these molecules were doing, because this is an orphan receptor. We were interested, given the high expression of ER alpha in skeletal muscle, and it is known to be a key regulator of fatty acid oxidation and mitochondrial biogenesis, mitochondrial potential. We then took, this is human skeletal muscle myotubes. We simply take, these are derived from primary cells derived from patients, and we can differentiate them into myotubes, from myoblasts into myotubes. We can then treat those myotubes with tool compounds and then use this really cool probe. There's a series of these probes called mitotracker probes. These are probes that will selectively get recruited to the mitochondria based on on the mitochondrial potential, and it's roughly proportionate to the mass of mitochondria in a particular cell. So here's a classic myotube, human skeletal muscle myotube, nuclear hoax staining. This is Floyden staining of actin filaments, and you can see only a very little amount of red staining from this mitotracker dye, paranuclear, just outside the nucleus. But when we treat these compounds with our ER alpha compounds, treat these cells with our ER alpha compounds, you can see really intense staining outside the nucleus. And we've used this along with some other data. I think I have a slide. So what this shows is that these are potential molecules for diseases of muscle metabolism, such as diabetes and other mitochondrial diseases. And we've used, because we didn't quite understand exactly how these mitotracker dyes work, and it took a number of other experiments to really validate that we were in fact upregulating mitochondria in these muscle cells. And this is one example using C2C12 mouse myotubes. And this is a nice system to use, a nice subunit complex to use. This is complex 4, cytochrome C oxidase. The nice thing about it is it's got several gene products from both mitochondria and also the nucleus. This is a Western blot with increased treatment of ER alpha tool compound. See that we see an increase in subunit 1, which is a mitochondrial encoded gene, and an increase in subunit 4, which is a nuclear encoded gene. We're also interested in one of the gene products we're interested in is CPT1B, which is the predominant importer of fatty acids into mitochondria. If we're increasing mitochondrial biogenesis and mitochondrial potential, you expect an increase in fatty acid uptake in OXPHOS, and we do see that with CPT1B. And this is relative to control, of course. So where we're at now, we're in animal studies, and we think that if our hypothesis and our cell-based data holds true, that these kinds of ER alpha modulators can be of value in diseases where reduced mitochondrial capacity or reduced mitochondrial function is the underlying cause, such as obesity, diabetes, non-alcoholic fatty livers, even Alzheimer's if we can get to the brain, and heart failure, and many other diseases. And interestingly, what we found with this is that, and there's another class of receptors from a competing company, a class of compounds from other companies such as Merck and Novartis, that showed that those compounds downregulate receptor in muscle cells, such as muscle cells. Our compounds apparently upregulate in a very dramatic fashion, even more than what I showed with some of our estrogen ER alpha compounds. Our tool compounds tend to upregulate ER alpha quite dramatically in C2C12s, and we think this is perhaps a reason why we're getting this agonist effect of these tool compounds. Modulation not only consists of how the ligand interacts with the receptor, but what the ligand does to the receptor abundance in that particular cell. We think here that these ligands stabilize the amount of receptor in muscle cells. So my final slide here, and the only reason I'm showing this is because of my work here back at Richmond in Dr. Topham's lab, which was dealing with iron-containing proteins, the enzymatic release of iron from ferritin, and just so happens we were involved in this project in collaboration with Mitch Lazar at UPenn that showed through a series of experiments that this sporphin receptor Reverb alpha, this is the LBD expressed as a wild type LBD, and this histidine mutation, when we purified the wild type receptor, it was orange. Okay, so that implied that perhaps it was binding something such as iron. This is purification work done by Bruce Wisely. Work with our analytical group showed that in fact it did contain the perfect mass for heme porphyrin, and in collaboration with Mitch Lazar, he's done a really, since discovering this, done a really elegant series of experiments using siRNA knockdown and overexpression studies and some mutagenesis as well to show that heme can bind and regulate RevERB alpha, interact with this critical regulator of circadian rhythm called BMAL1, and also interfere in the gluconeogenic pathway. So we think it's, based on some of our data and his data, we think it's perhaps a good target for diseases involving gluconeogenesis and overall metabolic diseases, and also perhaps recent data suggesting it could be of value in asthma. This is the first demonstration that heme regulates the nuclear receptor, and we hope that in future work, by finding new ligands to the receptor, either with or without heme, that we can make some progress with this orphan in some new diseases. So just to wrap up, I hope what I've showed you is that the NR-LBDs are really plastic, flexible, dynamic domains, both inside and outside the pocket. They're able to adapt to multiple binding partners. That's the modular hypothesis that we can alter that interaction network with ligands. They're well suited, we think, for drug discovery. One of the nice things we are trying to hold on to at GSK is this structural class systems-based research effort, where we have a number of scientists working in a particular area, such as nuclear receptors, such as 7TMs, such as kinases, where we can focus and have our basic research in addition to drug discovery research. And multiple advantages for that kind of a setting. For GR, new sortal ligands in the clinic. I didn't mention much about the novel non-sortal molecules. There's also some potential there. For ER alpha, I showed one novel modulator. We hope there's some utility for these compounds down the road for menopausal symptoms, osteoporosis, and cancer. And two orphan receptors. I told you about some of our more recent work, ER alpha and RevERB alpha. Finally, some work spearheaded by Tim Wilson, which I think is a very noble effort, and I hope will benefit the entire scientific community in the future. And that's the idea of an open access scheme, where molecules like we've discovered can then be made immediately available to the public and to the academic community for target validation work and pioneer drug discovery. So we have high hopes that this will be a kind of a new avenue for us in the future. So to wrap up, a great thing about being at a place like GSK is the opportunity to work with great scientists and work on teams and multidisciplines and all come together with a common goal. And there are many people to thank here, but these are just a few of the folks. I mentioned a few names throughout the talk that have come together to create these successes. And Eric Zhu, who along with Bob Nolte saw the original GR structure, is now at Van Andel Research Institute. So with that, I will close and take any questions if there are any. And thank you again very much. Pleasure.